oh, look at that. It's a cat's. That doesn't make sense, right?
It's a fish.
So, alumnus, singular.
I never knew that.
Alumni, plural.
Where does the word come from?
I think now we have to look. I mean, it's Latin. It's obviously Latin because the us-I ending is a Latin ending. And this is probably just a straight Latin word. Would be my guess. So an alumnus is just somebody who has graduated from that school, right?
No, I wonder what it means.
So let's look at the etymology of the word alumnus. But again, I think it is just directly Latin. It is from the Latin alumnus, a pupil, literally foster son, a vestigial present passive participle of alere, to suckle or nourish, to grow or to nourish, with the ending akin to Greek omnios. Plural is alumni. The feminine form is alumna, a plural alumnae, but that is largely not, like that would be true in the Greek, but not something we do in modern English, right? Like we don't tend to genderize our terms. We still do once in a while, like waiter, waitress, steward, stewardess, but that's becoming passe, right? We're actor, actress, but that's sort of becoming...
Waiter. I just call everyone a waiter.
Yeah. You call everyone a waitress if you want. I don't know. Anyway, so there you go. Now we know. It means to suckle, or it comes from a verb which means to suckle. And doesn't that just make you feel better about it? How does it feel to be suckling at Eastdale Secondary School right now? Would you say you're suckling here or just sucking? Anyway, let's move on.
All right.
I'm so glad I'm recording this right now. That's great. Yet another red letter moment in Joe Orlando recording his lessons. As you recall, this is going to be total review from last year, but I want to focus it on what we now know about the cell. So maybe we'll learn a little bit as we go. One of the central beliefs of cell biology, or biology, is cell theory, and it states, as we know, all living things are made of cells. And where do cells come from?
Other existence.
Exactly. All cells come from previous generations of cells. Now, obviously, this gives us a problem because cells haven't always existed. So this is fundamentally a modern theory, right? It relies on cells already existing. But our basic understanding is that our current best understanding is that the cell itself evolved once. Interestingly, in the reading I gave you on your last test, you saw that being eukaryotic actually evolved a few times separately. That's what our most current research is telling us. And by the way, when I say current, that article was about a paper that was published like the day before you wrote that test. Like I found a nice up-to-date article, which I thought was kind of fun. But in fact, I found about 7 articles, and I kept changing my mind about which one to give you, and I landed on that one. I liked that one. Hopefully, you guys did well on that section. That was the best done section on the test. So hopefully that means you thought the article was interesting. Or it just means that you're good at English class. It means you English real good. And I'm here for it. Which, again, I get, because English, as we know, is a good time. So, keeping in mind that the DNA or potentially RNA of the cell is the material that contains the blueprint for the cell, the reproduction of that cell must include the reproduction of the DNA or RNA. All right? So, believe it or not, before reproduction, the DNA is duplicated. And so what you'll literally see in the cell is that we go from a nucleus with a bunch of chromosomes to a nucleus with a bunch of chromosomes that now look like this. And the reason they now look like that is that they have duplicated. I should say replicated. That's the term we actually use. Not replicated. That's not a word. I'm struggling mightily. Guys, I'm struggling. There we go.
Are you not going to use different colors like you normally do?
Well, I will. Don't worry, I will. And if we zoom in on that, what we can actually see, it's actually kind of neat. Remember we said DNA makes that kind of double-stranded helix? Like that? Well, what we actually find is that what happens, and you'll learn more about this next year, is that DNA actually unwinds. And a second copy is built right alongside it like that. And then it rewinds. And what you end up with is 2 copies of the DNA just like that. We'll learn more about the exact mechanism of that in your grade 12 year if you take grade 12 biology. So you don't have to know the specific details, but it's actually really neat. The mechanism by which DNA replicates is that your cell actually contains, DNA that is a combination of an old copy and a new copy, right? We split it in half, we build the other strand right alongside of it, and we make a copy of it. So this is what it looks like at the molecular level. You don't have to know that for this course, but I think it's kind of useful to. Does that make sense? The big thing to know is at the chromosome level, the chromosome now looks like, well, that was fascinating. The chromosome now looks like this. It looks like 2 copies of itself, and it's held together at a central structure called, does anyone remember? Called the centromere. This is still considered a single chromosome. It's a duplicate or a replicated chromosome. Does that make sense? So this is still considered a single chromosome, but it's replicated.
Is it near or sphere?
Centromere. Yeah. So this is a chromosome, and the chromosome, once it's been replicated, is stuck to a copy of itself. You can almost think of it like, imagine if you photocopied a piece of paper and then stapled it together. Does that make sense? You can think of the centromere as being like the staple, and you can think of this being like putting it right in the middle of the page, which obviously only a freak of nature would do. Abdullah, who put his staples in wild and crazy places.
I see the one that you knocked Mark off.
Yeah, I took like 10% off all of his assignments because his staple was in such an obnoxious spot. It was like here, like unacceptable. Like it was way down here. So I was just like, if I have to take your staple out and put it back in here, I'm taking 10% off your grade.
Can you just bring back out?
Yes.
He probably was so mad.
He was not happy.
That was bad. That's valid.
I stand by it. But he was not happy with me. We had words, but it was okay. We got through it. We were stronger in the end. But yeah, he was unpleased. So, in asexual reproduction, all of the DNA is perfectly copied, big asterisks, except when it isn't. And then the cell divides. So if we look at the cell life cycle then, we have the regular portion of the cell's life is called interphase. Interphase itself will often be broken up into the sections G1, which is just kind of its growth phase, and then the S phase for replication, and then G2. That's not as fancy as it sounds. Basically just growth, DNA replication, and then more growth. Basically, if a cell is going to reproduce, and not all cells do reproduce, some cells stay in interphase forever, in which case they don't ever leave G1, they just kind of do their thing. Most of your specialized cells in your body are an example of that. Your brain cells are never going to reproduce. Your skin cells are never going to reproduce. We have cells whose specific job is reproduction. So we're talking here about a cell that is going to reproduce. Does that make sense? So it grows, and then at some point it's going to have to replicate its DNA, and then notably, it doesn't then split right away. It'll kind of keep growing. It'll do that for a while, just having 2 copies of the DNA. Turns out that's fine. A cell can be sitting on 2 copies of the DNA for a while. That's totally cool. It's just got twice as much DNA, but they're exact copies. Does that make sense? So now your 46 chromosomes, every single one of them would be basically like stapled to a copy of itself. Still 46 chromosomes, but twice as much paper. If DNA were paper, which it's not. Shut up. And then after interphase, then we enter the next stage, which is known as mitosis. Oh, sorry, I had a whole theme here, which I was doing this thing. Mitosis. And mitosis is where cell division happens, right? And of course, you probably remember the stages of mitosis, even if you don't remember what they mean. Prophase, then metaphase, then anaphase. This is not fitting well. I'm going to try again. Guys, I'm going to try again. I failed you. Metaphase, then anaphase, then telophase. And then the last step is cytokinesis, where we actually get 2 cells. We'll draw it, but does that make sense? Now again, this is asexual reproduction. We're making a perfect copy of the DNA, and then this is all about the cell splitting its assets, and then this is where the cell actually splits in half. You can imagine that if you were going to take all the stuff you owned and divide it between 2 houses, first you'd have to like pack a bunch of stuff up in boxes and move it around, right? And then, load up your moving van, do all that jazz. That's what mitosis is. Mitosis is all about moving stuff around and putting it where it's supposed to go. Am I good to go? Do you need a minute? I don't want to rush you. A minute? Okay, take your minute. You can take 2 minutes if you want. Don't let me rush you. I'm going to get myself a juice box.
I mean, a lot of pain between losing legs, then a lot of pain between my freaking legs. Because I did that yesterday.
Mr. Linda, you work in a restaurant, yes? Is this the proper way that fancy, like you're actually supposed to set up like a table?
Well, yes and no. Like every place has their own place. But yes, in general speaking, always knives on the left, always forks on the right as a rule. Soup spoons typically go on the left. Teaspoons often will go up above or maybe on the right. There's a whole thing. And then when you're dining, you just work your way. It's actually easy. Never be intimidated. You just work your way from the outside in. So like your salad fork, if your salad course is first, is going to be the furthest out fork. So you just grab that guy and eat. And of course, the reason the fork is on the left is you're supposed to hold the fork in your left hand and the knife in your right hand, right? And then that's why the soup spoon is on the right, because you use your right hand for that. Of course, you're supposed to scoop away from yourself, not towards, right? And then, anyway, yeah.
I flip it because I'm more comfortable.
I should have better manners than I do. When I was a young boy, my grandmother used to give my sister and I prince and princess lessons and teach us how to eat like properly. This is very important to her. She's very British, right? So we had to know how to eat properly, right? My father-in-law, who ironically is like a farmer from Vineland, he had dinner with the Queen once. And it was during the 80s. So he said that you weren't allowed to stop eating before the queen, and you weren't allowed to smoke until the queen had finished eating. And this was in the 80s when like everyone just smoked in restaurants and smoked inside at dinner. So he said it was one of the funniest things I've ever seen, because when the queen finally put her plate down and was finished eating, all of a sudden like 200 cigarettes came up and like everyone started smoking. But yeah, I have not dined with the queen nor the king. Okay, so now we're going to take a look at this. Most eukaryotic cells undergo the process called mitosis, and this should be a review from science class. So we just want to remember that in eukaryotic cells, DNA forms into chromosomes. We talked about this yesterday. We talked about it on the previous page. I just want to review it, which is that our genome, the human genome, is split into 23 chromosomes. We are diploid, which means we have 2 copies of each. And that means that we have 46. I can't stress this enough because it's an easy mistake to make. Don't draw this, okay? Look at me. Just put your pens down for a sec. It's fine. It's not going anywhere. It's not going to just look at me. So I want to be clear. When you see this has nothing to do with diploid. Does that make sense? This is not 2 copies of the chromosome, one from mom, from dad. This is after your own cell has made a copy of its own chromosome. Does that make sense? So that means if you look, you have chromosome 1, you have a copy from mom, a copy from dad. Chromosome 2, you have a copy from mom, copy from dad. Chromosome 3, you have a copy from mom, copy from dad, all the way up to chromosome 22, where you have a copy from mom and a copy from dad. And then we're going to assume for the purpose of right now that this person is female, which means that they're going to have 2 copies of the X chromosome. All right? And this is what they look like. So this is 46 chromosomes before replication, all right? So during interphase. In other words, when the cell is just living its cell life, when it's just doing its thing, this is what's inside the nucleus. Does that make sense? 46 chromosomes, 1 through 23, paired off. Does that make sense? And we can say, one is maternal, the other is paternal. What's that mean again? Father and mother. That's right. In other words, my chromosome number 1, in every one of my cells, I have a copy of chromosome number 1 that came from my father's genes, and I have a copy of chromosome number 1 that came from my mother's genes. And I have a copy of chromosome 1 from my mother, and a copy of chromosome 2 from my father, a copy of chromosome 3 from my mother, and 3 from my father. In my case, I have a copy of my X chromosome from my mother, and then obviously my Y chromosome must have come from my father, because my mother doesn't have a Y chromosome, right? Does that make sense? If you're female, you have 1 copy of your X chromosome from your mother, and 1 copy of your X chromosome from your biological father. Sorry? Like up here? Oh, out there. Do you want me to go yell at them? Do you? Okay.
Thank you.
He's a nice kid. Be a shame if I had to throw a stapler at him. Yeah, you bet. I think they are up on my lectern. Yeah, I think it's up there on my lectern. Is that true? Is that it? I think, because Sunena handed them out, so I'm not actually sure where they are.
They could be in the handout folder.
They might be in the handout folder. The actual lesson. Yeah, check the handout folder. And if it's not there, ask Sunena. She'll be there. Okay, and then after replication, what does this look like?
Are we supposed to draw that?
I mean, supposed to. It's up to you. But
said we were.
oh, that was this over here. Sorry, you can draw this. You don't have to. Well, hold up. You don't have to. What's important to me is that you understand this. All right? If drawing it helps you understand it, wonderful. If you understand it without drawing it, also wonderful. I would draw it, but that's totally up to you. Like, to be clear, for all my notes, like, you can write what you want. Like, I'm not grading your notes, but I try to write what I think is useful for you. Some people find it helpful to write a little bit more, and some people find it actually helpful to kind of cherry pick what they think is most useful. I make what I think is my best version. So now, after replication, you have 2 copies of chromosome 1, right? That looks messy. What if I did this? Is that better? That's a little better. Now we have 2 copies of chromosome 1, and then we have 2 copies of chromosome 2. I just really want to get that idea across that when you see this little X, that's not the 2 copies from mom and dad. That's a single chromosome that has been duplicated, and then they are joined at this structure. And remind me, what's that structure called? The centromere. Good for whoever said that. And if nobody said it, because I didn't actually hear anyone say it, then it's the centromere, guys.
Alex said something.
Excellent. It's one of many reasons I love Alex. Yeah, you bet. Does that diagram make sense? I have tried for years to think of the clearest way to present this information so that we don't get mixed up. Because when you think about it, it's weird. When you're like, you have 2 copies of every chromosome, and then they make a copy of themselves. And you have to think, like, does that mean we have 4 copies? And it's like, well, kind of, but they stay stuck to themselves, and they don't, you know what I mean? So you still have 2 of each chromosome. It's just before you have 2 of each chromosome, and they're just there for your cell to do its thing. And the purpose of this is to be split apart. This is in preparation of tearing the cell in 2 and making sure we have 2 cells and that they both have their own copy. Does that make sense? This is like making a photocopy, putting a little staple in there so it'll stay stuck together so that later on you can take the staple out and say, okay, you take a copy of this paper and you take a copy of this paper. It's kind of like this. If you ever sign a document, like if you get a mortgage or a loan or something, you go to the bank and they make 2 copies of every document. Why? You're going to sign both of them. Why do they make 2 copies of every document?
So they can keep one.
Well, the bank's going to keep one, and
You can keep one.
you're going to keep one. That's kind of the equivalent of this. We're making this copy here so that when we're ready, we can split the cell in half, and this cell can have one of everything, and this cell can have one of everything. Does that make sense?
That's what happened at camp. I just signed 2 copies of my contract.
Mm-hmm. I don't want the little kid thing to happen where it starts making sound, but I also don't want to waste the last of the juice, so you guys might.
I can cut the top open.
Because I want some juice. What kind of a question is that? Why in the world else would somebody have a juice box? Like, what are the list of reasons that people get juice boxes? Drink juice comes to mind, and then I'm really struggling to find the other reasons.
Sugar intake. Is sugar not part of it? If their parent forces them to take it.
Okay, parent forced me.
There's
That's
some
fair.
people, if they need it, if they're
Jack, mom did not ask me if I've drank my juice today.
Is your blood sugar low?
Though my mom did text me and say, drive safely. I am 42. I'm 42 years old. I've had a driver's license for, so what's that, like 25 years now? And like, if it is rainy, my mother will still text. So yesterday, my mom was like, make sure you drive safely. It's very rainy. And then today, my mother texted me and she's like, drive safely. Just because the roads are nice doesn't mean there aren't hazards. She's like, thank you, Mom. I appreciate that. Anyway, yeah.
I still want your cups and go get some water.
Sure. Yeah, you bet. Okay.
My parents.
So, mitosis preserves the number and information in the chromosomes from parent cell to daughter. So, when the time comes, both new cells get a copy of each chromosome. And the result is clones. All right? The daughter cells are clones of the parent cell. They're exactly identical. Just like if you pay that Chinese company to clone your dog. Have you heard about this? There's a company in China and they will clone your dog for you, but it costs like $200,000. So if your dog has passed away and you have like 200 grand, you can send them like a hair follicle and then you can get like your dog again. But it's not like it would have its memories or anything. So it's still just a different dog, but it'll look just like your dog. So that's kind of cool. And it should, in theory, have the same temperament. In theory, I don't know. The old nature versus nurture of it all. I don't know how I'd feel about that. Like, I love my dog, and like, obviously, I'll be sad when my dog passes away. I've been so sad when all my dogs pass away. But I feel like it'd be weird to be like, okay, replacement dog's here. You know? I don't know.
It's like when you have a goldfish as a child and your parents.
I had the opposite problem. My son had a goldfish, and then he was a goldfish slumlord. His goldfish tank was so gross. It was so dirty. Like, he took such bad care of it, and the things just would not die. They lived for, like, ever. And then we finally got, like, a nicer tank and this or that, and then, like, we took care of it. It still would not die. And then finally, like, my son went to the University of Toronto, and I didn't want to take care of fish. So I gave them to my sister works at a Southridge homeless shelter in St. Catharines, and it's still there. And these goldfish are, like, they're old. They're like elderly. They're like geriatric goldfish, and they're still kicking. And it almost makes me wonder, like, did they keep dying and he kept replacing them? Like, was he doing the opposite with us? I don't know. But
Not a goldfish, it's a beta fish.
yeah.
It was my client that I used to see. She passed, and it's still alive.
Wow, that's kind of adorable, though.
It's like crazy.
But just one?
Yeah, just one.
Oh, wow.
Beta fish can't live with two. They'll fight each other.
Oh, do they?
Unless
Oh,
they're the
interesting.
same sex.
No,
Then-
No,
males will fight each
opposite,
other.
sorry.
Opposite sex.
Yeah.
Opposite sex mates, same sex fights.
Yeah.
Got it. That makes sense. All right.
But it's hard
Because
to
the
know
opposite
if
wouldn't
it's
be
the
very
same
good
gender
procreative
or-
strategy.
That's why we put like
I don't
little
think
guppies
females
with
are
it, so then they can always fight.
Okay, so let's take a look then. This you've already just seen. All right, you've already seen it. G1 phase, S phase, G2 phase. I don't need to say anything new here. I don't know why I left that big space,
Great.
but it's there. You could draw me a.
Can I draw?
Yeah, sure. Please. You're good at drawing. My goodness. But I'm going to let you do that later because we are going to jump into it. I guess there's one thing I'm going to say, which is after, like, you can put it at the bottom here so you leave lots of room for your doodles. After DNA replication and a period of growth,
Is this under interface?
sure.
Why is
Where's...
It's just at the bottom of the page. It's just, there you are. There we are. Whoa.
all the way down there?
To leave you room to doodle, I'm being conscientious. I'm being conscientious. I'm a conscientious guy. So after DNA replication and a period of growth, the cell will then enter mitosis. The cell will actually begin dividing its assets. It'll begin dividing itself to split in half. For prokaryotic cells, that's quite trivial. It just has to move one copy of the DNA to one side and one copy of the DNA to the other side, and it can pinch in the middle and it can divide in half. What do we call that process?
Yeah.
Cutting into two. We call it? Nope. What? Functionally, yes. But when that's the only thing that has to happen, you're absolutely right. What do we call it? Cell division. More specifically, what type? Fission, specifically into two. So binary fission. Excellent. Good. Yep, you got it. So, but in the case of eukaryotes, there's more stuff to deal with. Does that make sense? You got mitochondria, and you've got endoplasmic reticulum, and you've got Golgi apparatus. We're not going to look into all that, but we are going to focus on the nucleus, right? In eukaryotes, there's a nucleus, and we have to deal with that because you can't just cut the cell in half because then one cell would have a nucleus and one cell wouldn't, right? So, how is this handled? And the answer is that the DNA, the chromosomes as we show them, don't actually become visible like that until this stage. So during prophase, at the beginning, there is what looks like just a tangled mass of DNA in the nucleus. And over the course of prophase, the nucleus itself is going to start to dissolve. The nuclear membrane is going to break down. And the chromosomes are going to form into their visible shapes. Now, there are actually, of course, 46. For the purpose of this whole diagram, I'm going to pretend that humans have 4, because I don't want to draw 46 chromosomes. If you would like to be extremely accurate, and you want to draw 46 chromosomes, hey, don't let me stop you. But this is what we're going to start to see. And we'll talk a little bit about here. Now, I want to be clear. Well, we'll get there. Give me a sec. I'm going to draw it, and we'll talk about it. 1, 2, 3, 4. Okay. And of course, we know that the centromere holds those together, right? And then these structures at the pole of the cell are going to form, and then they're going to start to shoot out these little tubes, these little fibers. So the nucleus is going to dissolve, and that's what we're going to show here. The nucleus dissolves. The DNA coils into chromosomes. And when we say coils, it really means, have you ever seen like an old telephone cord? You know, the ones that coil like that? DNA already kind of looks like that, right? Now imagine if you took that and you coiled it again. It would go even more compact, right? Then imagine you took that and coiled that. Then imagine you took that rope and coiled it. That's what actually happens to DNA. It coils 5 times. We call it, scientists had to think of a really clever name, and you know what they came up with? Supercoiling. The DNA coils, and then it supercoils to form the chromosomes. So the DNA coils into chromosomes, and then finally, this is called a centromere. Now, no, it's not. That's called a centromere. This is called a centriole. The centrioles form. Now, I wish that these were not called centrioles and the ones on the DNA were called centromeres. I think that's silly naming. I wish we called one like the DNA staply knobbly bit, and we called the other one the like spindle fiber producing knobbly bit. But we don't. We call them centrioles and centromeres. The centromere is the thing that holds the 2 copies of each chromosome together. And the centriole forms here. And then, once the nuclear membrane is dissolving, these will form, and these spindle fibers will begin to form. And it's kind of artificial what we call the end of one phase and another. So I hate when people like obsess with PMAT, as if that's like the most important thing. Prophase, metaphase, anaphase, telophase. Like, yeah, we give the phases names so that we can kind of see it. But like the phases are artificial, right? Like we just said, okay, this bit we're calling that prophase, and this bit we're calling it metaphase. The important thing is, why does this have to happen? What is the cell trying to do here? It's trying to go from being 1 cell into being 2 cells. So what's it have to do? It has to split. But before it can split, what does it have to move? Anything else it can make. Say it accidentally makes a cell and one of the 2 cells has no Golgi bodies. It can just build new ones. Say one side ends up with not enough vacuum, like lysosomes, whatever. It can just build more, right? But what do both sides absolutely need? They need DNA, right? So all that happens in mitosis, don't memorize it. Don't memorize it. Think about it. If you think about it, it's obvious, which is we're going to have to take the DNA and we're going to have to put half of it over here, and we're going to have to put half of it over there. Okay, well, if you were doing that, what would you have to do? Well, first of all, normally the DNA is held safely inside a structure called the nucleus. Well, you can't move the nucleus to this side because then there's none on this side. And you can't move it over here because then there's none over there. So we're going to have to get rid of the nucleus, which happens. Does that make sense? And then we're going to have to use something to move it. So I don't know. What if we use like basically ropes, right? That's how we often move stuff, right? That's basically what spindle fibers are. We say, okay, we're going to put a big anchor on this side of the cell. And then we're going to build a bunch of little tubes, a bunch of little ropes that are going to go out and grab the stuff and pull it in, basically fishing. And then because we want half of it to go to the other side, we're going to do the same thing on the other side, right? And then if you're moving something in your house, say you want to move your clothes, are you going to start grabbing like a shirt here and like a pair of socks there and a pair of underwear here and a sweater here? Is that how you're going to move your stuff? Of course not. You're going to take it and you're going to pack it into boxes. So we take the DNA and we pack it into chromosomes. Do you see what I'm saying? Like, this isn't something to be memorized. It's something to be understood because it's an engineering problem. The cell has to go from being one cell to being two. We got to get half the DNA over here, half the DNA over there. And if you think about mitosis that way, it's just the logical steps that have to happen. Yeah, we're going to have to get rid of the nucleus because otherwise, how the hell would you get the DNA to two different sides of the cell? Right? It's obvious. Well, we're going to have to move it. How do you do that? Well, we got to build something. We build little ropes. So the cell is doing something really logical. Now, what are these ropes going to do? They're going to grab onto the DNA. But they don't grab onto the DNA itself. They're not built to do that. They need to have something to grab onto. What part of the DNA is built for them to grab onto? The little knobbly bit, which we call the centromere. The centriole is going to connect to the centromere. And the thing that's going to connect the two is these little ropes, these little spindle fibers. And that's going to happen in our next stage, what we call metaphase. In metaphase, and we can really just draw this once. By the time metaphase is reached, what has happened is that the nucleus is totally gone, and the spindle fibers go out in search of DNA. They build all over the place, and they don't all necessarily find one. But what's going to happen is they will find the centromere of each chromosome. And they'll also send a lot of spindle fibers out that do nothing. Does that make sense? They just kind of go out in search of stuff. They actually move some stuff other than the DNA, but we're just going to focus on the DNA here. Is that fair? And from this side, the spindle fibers are going to go out and they're going to go in search of all the different stuff. But again, noteworthily, they're going to find each of the chromosomes and they're going to start pulling. Now, we could demonstrate this if we have to, but if you take something that seems to have two arms and two legs and you pull on it from both directions, at first, where's it going to end up? In the middle, like a little tug-of-war. Does that make sense? So again, you don't have to memorize this. It makes perfect sense. If you start pulling on the chromosomes from both sides, they're going to end up in the middle. They're going to end up in the middle. Does that make sense? Imagine if you and I went fishing and we both caught the same fish. Right? You're on that side of the pond. I'm on this side of the pond. We both cast our net. We both catch the same fish. It goes deep. Both our lines at the same time. And now you're reeling. Come on, reel. Come on. Give it. Yeah, there we go. That's good. Come on, give it. Like you care. You want this fish. I want this fish. I'm going to get this fish. We're reeling in. Where's the fish going to end up?
In the middle.
Closer to me, I'm stronger, but you get the idea. You get the idea. Anyway, but I'm tough. First thing people notice about me. But does that make sense? The fish is going to end up in the middle. So at this stage, where do the chromosomes end up? In the middle. But not for some random reason. You don't have to memorize that they end up in the middle. Of course they end up in the middle, because you're pulling on them from both sides. It's a tug-of-war. Now, I want to get away from the fish analogy, because the next step would be a little gruesome in that regard. But in for a penny, in for a pound. Let's say neither of us is really going to give up. No, I don't like this. Never mind. Let's say it's not a fish. Let's say that we have both caught a sandwich. Okay, sure. And we're both reefing on this sandwich. Eventually, if we pull hard enough, what might happen to it? It'll break in half. You can have half a sandwich, and I can have half a sandwich. There you go. You're a triangle guy? I respect that.
Yeah, you want to put them in a triangle.
You like the triangle? Okay, I respect that. I think it depends on the type of sandwich.
Oh, yeah, sure. Yeah, if it's a
Like grilled cheese triangle?
Yeah, triangle.
Oh, interesting. See, grilled cheese to me isn't a triangle. Club is a triangle.
Club's a triangle.
Yeah, BLT is a triangle. But I gotta be honest, but grilled cheese, I'm just right down the middle guy. Interesting. Squares. Oh, anyone a strip person? Like they cut the grilled cheese into little strips? I knew a guy. Does that make you a sociopath? Maybe, right? Anyway, let's focus.
Squares.
I've seen squares. I respect squares.
I like 4 triangles, not 2.
You like 4 triangles, but triangle, not square. Not square. All right. Now I want a sandwich. Guys, this has backfired horribly. Okay, anyway. Now, again, do you see how we're not memorizing any of this?
Yep.
We're talking about fishing for sandwiches, for God's sakes. But you don't have to memorize it. Just imagine fishing for sandwiches. You know what I mean? Like, sometimes you have to memorize things, but sometimes it's better to think about things as a process. And if you mess the names up, whatever. Like, that's not as important. Obviously, ideally be perfect. But really, in the end, it's steps that make sense. Does that make sense? I think getting all hung up on memorizing the names and this or that, the important thing is we want to show you that the cell has a mechanical solution to solving a problem. It has machines that it's built that does this thing. So now in the next stage, anaphase, we'll draw this one in 2 steps. The DNA is actually going to split in half because we're going to yank on those chromosomes until eventually they actually break. And I'm not going to draw all the spindle fibers to nowhere, not because they don't exist, but just because it becomes messy. But basically, the DNA is going to get pulled to the 2 poles of the cell. Right? Pulled to the poles. I don't even want to put words. Do I need to put words? I feel like we don't need words. Just the picture. The picture tells you the story of what's happening, right? And then, having done that, the DNA is important. It's precious. We don't want to get damaged. The nucleus was there for a reason, right? The nucleus was there for a reason. So what do we want to get back around our DNA as fast as we possibly can? We want a nucleus around it again, right? So even before we've made 2 new cells, as the 2 new cells are starting to kind of differentiate, we're already starting to form a nucleus again, right? We've got 1, 2, 3, 4, 1, 2, 3, 4. And by the time we are here, this is what we call telophase. But again, like, yes, I want you to know the phase names, like whatever, that's curricular, it's important. But I think the curriculum's so silly, because the curriculum is like, differentiate between prophase, metaphase, anaphase, and telophase. It's like, okay, like, I guess, who cares? Because those are artificial names we've put to the stages of the thing. But I would much rather you understand the process here, which is the cell is solving a problem, and it's doing it using a machinery, right? It's got machines that do the job and a fixed process that it follows. Does that make sense? Your cells are incredibly complicated machines, and they have all kinds of processes like this. This is just one example. And it's even, we're just scratching the surface. Our next lesson, you're going to learn how this happens for sexual reproduction, and it's even more complicated than this. But if you have a good understanding of this, the next one isn't so bad. Does that make sense? The reason I want you to have a good understanding of this is because when we then go to the next step and look at sexual reproduction, it's a variation of this. It's this with a twist. And so that's important. That's considered the end of God, what's wrong with me? No, mitosis. And then at the very last stage, we get cytokinesis, where the actual split happens, right? Where we actually have that cleavage furrow going all the way through the cell, such that we end up with 2 new cells, right? 2 daughter cells. And those daughter cells should be clones of the original parent cell. And then, not for nothing, the chromosomes should actually unwind again to just nice loose chromatin, because the cells can actually use that DNA to do the job, right? Like, it's going to actually live its cell life, do its thing. So that's my visual crash course reminder. Visual crash course reminder. I'm sorry, I can't speak English. It's not my, I think a lot about people. I'm reading a biography right now. It's a really good one. But it's just talking about Then this guy went and gave a speech in Germany in German. Then he went and gave a speech in France in French. And I just think like, boy, I can't even speak my own language such that I could give a speech. And that makes me sad. Anyway, that's the end of my story. How's that feel? Do we have a good kind of grasp of that, basically? Again, yes, I want you to know the phases, but much more importantly than knowing the phases, I want you to get the concept, which is cell has a task it has to accomplish. It does so following a strict process that makes sense. It's an engineering solution to an engineering problem. You got a bit of time. You can use it as you see fit. I trust you completely. That assignment I gave you is a good use of your time, but if you have something more pressing to do, don't let me stop you. That's very nice.
There's your drawing. I messed up your mouth, so you don't really thick beard.
No, I like that. Yeah, and also do memorize. And by the way, don't memorize, but do memorize. You should all memorize a poem today. Every one of you. Pick a poem, memorize it. Yeah, you. You could be like, I think I shall never see a poem as lovely as a tree. You know that one? You should look it up.
Can I use this?
Yeah, you bet. Yeah, verses are good. If you have scripture that you find sacred, a wonderful thing to memorize. But memorization is good for the brain and for the soul. Everyone should memorize.
So don't memorize.
But you shouldn't just memorize. Don't just memorize. Because memory is important. Listen, you will never meet a person, I believe this deeply, you will never meet a person whom you believe to be wise who doesn't also just know a lot of stuff. Right? There's more to wisdom than knowing stuff, but knowing stuff is a prerequisite. You gotta know stuff to be wise. So memorize stuff. Put lots of stuff in your brain. That's what it's there for. Even when we train artificial intelligence, how do we do it? Well, we have it read everything it possibly can, right? And then we have it try things. Can I see?
Show your art.
This is good artist. Yeah, I know. I should do that gym thing. Yeah. This is heavy. I should do it again. Lift it up again.
Yeah.
I'm saying this during my second series, but when you actually describe out loud what people do at the gym, what I do, I'm quite really fast, I don't go and stay right where I am. And then after that, I go on the side of the machine, I run really fast, but I don't get anywhere. I stay right where I am, I'm looking at a concrete wall, I smell the room surrounded by it. And I'm like, this sounds like every ancient description of going to hell. Right? Like if you read like ancient stories about like being punished for all eternity, it basically is describing having a gym membership, right? Like Sisyphus, what's he doing? Oh, push the wall up, push the rock up the hill, let it fall down, and repeat. That's a gym membership. You see what I'm saying? So for me, and I'm not saying for other people, for me, I'm good at this, not so good at the sporty movement body dance. That was never my specialty. You know, that's the stuff that they joke about. I was math was fine. Math was fine. But like basketball, that was rough. I didn't do so good with old basketball. With a hockey team for a year, and I was like, the kid with the like, oh god, they're about to put him on the ice. Now we're gonna get scored on it for sure. Yeah, that was me. I was like, yeah, it was awful. I'm just not an athlete, and I've come to. It made me sad for a while, and as an adult now I can enjoy playing sports and not being good at them. It's like it's okay to not be good at things. You can't be good at everything. Nobody's good at everything. Right?
Do these for like face masks? I have an allergy appointment, and they're like, make sure you bring a mask.
Yeah, you bet.
My mom doesn't have any.
Yep. Yeah, I got lots.